Okay, let's unpack this. Imagine a future where the body's own dormant capabilities could be awakened to fight disease. What makes this truly remarkable, and what we'll explore today, isn't just clever science, It's the paradigm shift it represents in how we might approach these really long-standing, devastating illnesses, the ones impacting millions globally. We're diving deep into a pretty groundbreaking new approach in gene therapy. It's called Delete to Recruit. And it's all about how researchers are using cutting-edge technology, not really to introduce new genes, but to precisely reactivate these backup genes that are normally switched off.
Exactly. And our mission for this deep dive is essentially to distill the key insights from this really recent research. It was published in the journal Blood. It's work from a collaborative team, Hubrecht Institute, Erasmus MC, and Sanquin. And it focuses on how this innovative method could offer some profound new hope for serious genetic diseases.
Okay, so to really grasp the implications, let's start with the problem this new method is trying to solve. We're talking about inherited blood disorders like sickle cell disease and beta-thalassemia. The core issue here is basically the adult globin gene malfunctions. It stops the body from producing normal hemoglobin, that crucial protein that carries oxygen in your red blood cells.
And the consequences are, they're severe and lifelong. Without properly formed hemoglobin, red blood cells just break down too quickly. That leads to debilitating anemia, chronic fatigue, and down the line, significant organ damage. It's rough. Many patients frequently rely on regular blood transfusions just to manage, which really raises an important question. What if there was another way for the body to produce this vital protein, maybe a different pathway that could be, activated?
Well, the good news is our bodies actually do have a sort of backup engine for producing hemoglobin. It's the fetal globin gene. This gene naturally produces hemoglobin before birth, powering us as fetuses, but then it's completely switched off after we're born. The challenge, obviously, has been figuring out how to switch it back on.
Right. So before we get into how they switch it back on, let's just quickly recap how genes work. You'll remember genes in our DNA. They contain the instructions for making proteins. But importantly, not all genes are active all the time. It's like a huge instruction manual where only certain pages are open at any given time. And crucially, there are these segments of DNA called enhancers. Think of them like switches. They turn genes on, and they can be located right next to the genes they regulate, or, and this is fascinating, sometimes much farther away along the DNA strand.
Okay, so how do they actually do this switching? This is where it gets really interesting. This is the delete to recruit method. Researchers are using CRISPR-Cas9, often called, molecular scissors.
Yeah.
But not to introduce new genes. They use it to precisely cut out a specific piece of DNA that sits between an enhancer and a gene.
That's exactly it. The mechanism is quite ingenious. By simply cutting out that intermediate piece of DNA, the enhancer is physically brought closer to the target gene. It effectively recruits it, turns it back on. So one of the study's first authors, Anna Felder, she explained it pretty clearly. She said, in the study, we discovered that it's possible to activate a gene by bringing it closer to an enhancer. And she also clarified, we directed the scissors to cut out a piece of DNA between an enhancer and its gene, bringing them closer together.
Right, and that's exactly where the name delete to recruit comes from. It makes perfect sense. What's truly remarkable here, I think, is the sheer elegance of it. The simplicity.
Yeah.
You're altering distance without adding any new genes or foreign material. It's like rerouting an existing electrical connection rather than installing a whole new circuit. Okay, now, bringing this back to sickle cell disease and beta-thalassemia. How does delete to recruit directly impact these conditions? The aim is to restart that fetal globin gene, right, to effectively repower the red blood cells.
Absolutely. And the significance of reactivating this backup, this fetal globin, it's profound. Anna Felder put it perfectly. She said, in people with sickle cell disease or beta-thalassemia, it's the adult globin gene, the main engine that powers red blood cells, that is broken. But fetal globin is like a backup engine. By switching it back on, we can repower the red blood cells and possibly cure these patients. And what's really fascinating here, the key finding, is that the strategy's efficacy wasn't just confirmed in healthy donor cells. Crucially, it worked in cells from patients themselves. And perhaps most importantly, in blood stem cells.
Wow, blood stem cells. It sounds huge.
Yeah.
Why is confirming it in blood stem cells so significant? Tell us more about that.
Well, it's hugely significant because these blood stem cells are the source, the factory for all the body's blood cells throughout your entire life. So if you can reactivate fetal globin in those stem cells, it could lead to a continuous lifelong supply of healthy red blood cells, potentially a functional cure.
The functional cure. That's incredibly powerful. Okay, so what are the hurdles then? Moving this from really promising research to something available in the clinic.
Right. Well, as with any gene therapy, even when this promising, there's a journey. It involves really rigorous safety testing, first and foremost. Then you need to optimize the delivery methods. How do you get the CRISPR machinery effectively into those stem cells? And then there's scaling up production. It's definitely a complex path. It takes time. But this research, it lays a phenomenal foundation. It's a really big step. And if we connect this to the bigger picture, the wider implications are really exciting. This method, this idea of deleting to recruit an enhancer, it could potentially apply to other diseases. Anywhere where an existing backup gene could compensate for insufficient amounts of healthy proteins. So it's potentially not just about blood disorders. That's what makes it so groundbreaking.
Right. So what does this all mean for the future of gene therapy then? Because there is actually a gene therapy for these conditions already approved, I think, in Europe just this year, 2024. How does delete to recruit stack up against that? What are the potential advantages?
Yeah, that's a great question. This new approach, it might circumvent some key issues with existing therapies. One big one is cost. Current gene therapies are incredibly expensive, which obviously limits accessibility for many people. Furthermore, the existing treatment works differently. It modifies a gene that normally represses fetal globin. And while that works to switch fetal globin back on, modifying that repressor gene, well, it might have unknown off-target effects on other genes down the line. We just don't know everything yet. The delete to recruit method seems potentially more precise because you're not altering the repressor gene itself. As Felder concluded, editing the distance to an enhancer instead of the genes themselves could offer a versatile therapeutic approach. It might be a path with fewer unknown consequences.
So, okay, to recap this deep dive then. Instead of trying to fix a broken engine, the adult globin gene, or replace it entirely, this team found a way to just flip the switch on a perfectly good dormant backup engine, the fetal globin gene that was sitting there all along, just needed bringing closer to its switch.
And this raises a really important question, something for you listening to consider. As gene therapy keeps advancing, how might our understanding of the human instruction manual, our DNA, how might that continue to unlock unexpected solutions? Not just by adding things or removing broken parts, but by simply repositioning what's already there. It makes you wonder what other backup engines might be waiting inside us, just waiting to be switched on.
Yeah, exactly. So the next time you think about genetic diseases, maybe remember that sometimes the most profound solutions, they aren't about introducing something entirely new. Sometimes it's about rediscovering and reactivating the amazing capabilities already hidden deep within us.